In this video, we are going to talk about fermentation. We've been talking about carbohydrate catabolism, and we're saying different kinds of bacteria can do it aerobically, so can humans, and we call that cellular respiration. Other microorganisms do it anaerobically, and we call that anaerobic respiration, so some organisms can do that. And then there's another anaerobic way that carbohydrates can be catabolized, and it's called fermentation. So the first thing to remember about fermentation is it occurs in the absence of oxygen, just like anaerobic respiration. So you have a diagram. So we're going to, you have some diagrams in your notes, and we're going to look at that. My first learning outcome, I have here, give a general description of fermentation, the kinds of organisms that do this, and some common end products and their commercial value. I just like this story. It's an important story in microbiology. There's a microbiologist named Louis Pasteur, and I think you might have even heard of him. He was one of the key people in the discovery of microorganisms and the germ theory that microorganisms cause disease. And he was doing all sorts of research. Well, the middle of 1800s, we didn't have microbiologists because people didn't really know that much about microbiology. But he was a chemist. And he actually discovered the process of fermentation in the mid-1800s, 1857. And what happened is he had a merchant that came up to him, and he said, I'm trying to make wine out of beet juice. They made wine out of everything in those days. So he was making wine out of beet juice. And what he found, and you know wine, what we're trying to do is we do fermentations and we make alcohol. And so sometimes his beet juice, could get alcohol out of it, but other times he got vinegar and it ruined it. And so the question is how can I consistently get alcohol and not vinegar? And they didn't even understand why. They didn't even know the mechanism of how to make alcohol. They knew that you took beet juice, or you took different kinds of fruit juice or vegetable juice, you put it in a sealed container, you left it at a certain temperature for a certain amount of time, and you got wine. But they didn't know how that happened. And so Louis Pasteur started studying this. And what he ultimately did is he found that microorganisms caused the production of the alcohol. And people didn't know that. They could look at wine under the microscope and see this. And these actually are yeast. You might recognize they're budding off. They're relatively large. And so these are yeast. And if you look at wine, you see yeast. And but the question was, some people said, oh yeah, you see yeast, but does wine cause the production of yeast, or does yeast cause the production of wine? So there was a lot of questions about, what are the yeast doing and why are they there? But Louis Pasteur did some pretty some elegant experiments that are historically significant, and he was the one that figured out that the yeast were actually producing the alcohol in a process that he called fermentation. And it was at this point in 1857 that people started looking at this and going, oh, so if microorganisms are changing food, they can make grape juice or beet juice into wine, maybe they're having impacts on the human body. Now, this wasn't that long ago, 1857. We did not know that microorganisms cause diseases. And so I have the question here, it says, why was the discovery of the mechanism of fermentation important enough to be identified as the starting of this important period of time called the Golden Age of microbiology, where we really started learning lots more about microbiology? And the point is that by understanding that yeast caused the production of alcohol in wine, or yeast was able to change beet juice into wine, that they may very well be changing a healthy body into a sick body. And it was at that point that people started understanding that microorganisms cause disease. So this was a really big time in the history of mankind, really. Okay, so we want to talk about fermentation. And you have the diagram that I gave you already. And you notice we've been talking about respiration. We talked about aerobic respiration and anaerobic respiration. And now we can go over here and talk about fermentation. I also gave you another picture that looks like this. These two things are the same thing. If we look right here, fermentation does not use oxygen. Remember, in aerobic cellular respiration, the oxygen's down here. And we don't go down there. We're just using glycolysis in fermentation. So oxygen's not required. And here we go, glucose to pyruvic acid. We have glycolysis in fermentation. Remember I told you in glycolysis we just make a little bit of ATP. We don't make a lot. Okay? And so when things grow by fermentation, they're going to grow more slowly than if they grew aerobically in the presence of oxygen, because you're only making a little bit of ATP. So in fermentation, we have glycolysis, and here you have glucose to pyruvic acid, and that's the same thing here, glucose to pyruvic acid. We're making a little bit of ATP, a little bit of NADH. But it doesn't stop there. You end up, depending on the kind of microorganism, you might end up, and here it's showing you can make some other things, and we call these fermentation end products. Some microorganisms can make things like lactic acid. And lactic acid is one of the primary bacterial products we use to make cheese. Other bacteria and even yeast can metabolize the pyruvic acid and make ethanol. And they actually make a little CO2 also. But they make ethanol. So depending on the kind of microorganism you have, your fermentation end product will be things like lactic acid, ethanol. There's other things too. So let me just back up. We use fermentation. The microorganisms can use fermentation in the absence of oxygen. Not all organisms can do this. So you have to have these enzymes that allow you to carry out these metabolic pathways. They can also grow by anaerobic respiration in the absence of oxygen, but most bacteria are not able to do that. So fermentation is just another option for those microorganisms that need to grow anaerobically but can't do anaerobic respiration. And at the end of fermentation, we have these different metabolic products. And a lot of these we've been able to shape into commercial products. Here's some examples. You do not have to memorize any of this, but here, just alcohol. If we want to, there's a pathway where you can produce alcohol through fermentation. We make ethanol, carbon dioxide. Here's some examples of, these are different kinds of yeast. And what do we do with that? We make beer, we make bread. There's a metabolic end product called butyric acid. Some bacteria can make that. And this bacteria called Clostridium butyricum. And it contributes to making butter and buttery flavors. Lactic acid is a product of Streptococcus and Lactobacillus. Go look on your yogurt containers. You're going to see Streptococcus and Lactobacillus in there because they're put in there to cause the milk to make these products. And so things like sauerkraut, cheeses, and yogurts are used for making lactic acid. So there's all these different kinds of, here's a good one, propionic acid, made by certain kinds of bacteria. It's used to make Swiss cheese. And the holes in the Swiss cheese are gases produced by the bacteria that are in the cheese making process. So we've used fermentation in a lot of situations to make commercial products. This table just summarizes our three pathways of the three ways that we can catabolize carbohydrates. We had aerobic respiration, where we get a lot of ATP. Our final electron acceptor is oxygen in the electron transport chain. Anaerobic respiration, there's a bunch of different kinds of bacteria doing that. Remember, we have something other than oxygen at the end of the electron transport chain. And these will grow a little bit more slowly depending on the kind of bacteria, because they're going to make a little bit less ATP, a lot less or a little less, depending on the situation. And finally, fermentation, and they gave you the example of a yeast here. They don't have an electron transport chain, so they don't have a final electron acceptor, but they do have, they only make a very little ATP. So bacteria and yeast that are growing by fermentation are growing much more slowly. Now, my last thing I want to talk about is look at your lecture study guide. I gave you some tables and things to fill out, some questions to answer. Looking over those and practicing those will be a really big help to understanding all of this. I highly recommend you practice the bacterial mega matching. That is like, if you can do the bacterial mega matching, you are getting the points that I want you to understand about aerobic respiration, anaerobic respiration, and fermentation. Concentrate on that. Make sure you don't just memorize that, but you understand it. Look at those pathways, compare these three things, okay? All right. From that point, actually, I'm going to tell you one more thing. Humans do a little fermentation. We make a little lactic acid. And think about that. Where do we make lactic acid? Oh, we make lactic acid in our muscles when they're wearing out. Why do our muscles make lactic acid when they're wearing out? Because they're running out of oxygen. So we can't do anaerobic respiration in our cells. We can do a little bit of fermentation, but it doesn't last very long. Our muscles don't work very long once they start burning from that lactic acid because we're only making a little bit of ATP. Okay? So humans depend mostly on aerobic cellular respiration, but in times of muscle stress, we do a little bit of fermentation and make lactic acid. So you've heard of this before. Anyway, good luck with all this. Take care.